Okay, in this video we are going to examine nitrogen balance. The primary feature of nitrogen balance we're going to be examining is the processing of amino acids into either proteins or energy. So let's start out in the middle of our page here with our familiar amino acids. Now we've examined the structure of amino acids in previous videos, but let me put this here just to remind everyone that what we have is an alpha carbon in the center of the amino acid, and bonded to this alpha carbon is a primary amine, nitrogen group, and a carboxylic acid. The important characteristic of amino acid, of course, is the R chain. This is the side group that gives the character of the particular amino acid. Now, amino acids are going to be coming from two sources while we're discussing nitrogen balance. The first source, of course, is going to be from the diet. Dietary proteins, recall, are processed by hydrolytic enzymes in the pancreas. So these particular enzymes are specific for proteins, and therefore they're part of a class called proteases. And recall, these are hydrolytic enzymes, and so they are designed to add water across the peptide bond and break the proteins down into their amino acids. Now, the amino acids under these conditions, because we are in a fed state, recall the fed state is going to be what is called anabolic. The anabolic state means that we are now in a building phase. Because we're in a fed state, we have high insulin levels, and this is telling the body that we are in a time of plenty. And so the amino acids, the primary pathway for amino acids in the fed state, is going to be the anabolic pathways, and we are going to assimilate these into proteins. So cells will take up the amino acids, and being in an anabolic phase will assimilate these into proteins by protein synthesis. Now, what I'd like to talk about mostly in this particular video is the concept of amino acids being used not for anabolic reactions, but rather for catabolic. And the catabolic reactions are going to be occurring in the fasting state, and the reason we are doing this is we are going to be using these amino acids to generate energy. Now, under these conditions, amino acids have to come from a particular source. This particular source, of course, is going to be protein, endogenous protein in the body. The most abundant protein we have in the body, of course, is in our muscles. And so muscle protein is going to be broken down into amino acids, and then once we're in a catabolic state, we will shift these hydrocarbon molecules into the energy-producing pathways. Now, how does this happen? Recall that amino acids can be used for both protein synthesis and energy in muscles. And so if we are going to be doing protein synthesis, this is going to be an anabolic reaction. The converse, protein degradation, this is, recall, protein half-life, and all proteins have a specific half-life, which means that there's a longevity to every protein in muscle. So when proteins reach their end of their life, they are broken down by proteasomal reactions. And the proteasome is basically a subcellular organelle that is designed to break down proteins that have been targeted for degradation. Breaking down of proteins, of course, means hydrolyzing the peptide bonds back into amino acids. Now, this is a catabolic state. When we get into anabolic, we can shift these amino acid pools back into protein to make new protein to replace those that we've degraded. Now, what happens when we get a little farther out on our energy utilization scale? So let's look at time versus energy used. Now recall, at early times following feeding, our first energy source we are going to be using, being in a fed state, is going to be glucose from dietary sources. So once we ingest the glucose in our diet, we are going to be using this energy until it's depleted. Now once this is depleted, we are going to be transitioning into the fasting state, and then we are going to be using glycogen for our energy stores. But recall that glycogen only has a specific amount of storage capacity in our body. So once we run out of glycogen, then we're going to have to be shifting to an alternative fuel source. This, of course, will be fatty acids. This fatty acid oxidation. Fatty acid oxidation, of course, generates a lot of energy, and this is going to be our main energy source in a long time fasting condition. Now, if we take this time out considerably longer, so that we are approaching a starvation state, let's say that our fatty acids are insufficient to supply us with the amount of energy we need, then we're going to rely on our final energy state, storage, energy substrate, which is going to be amino acids. And at this point, amino acids will be broken down because we are extended into a fasting state, which is a catabolic state. And we need a mechanism to increase the pool of free amino acids to be shifted into energy, rather than shifting into protein synthesis. The way we increase the amount of amino acids available for energy, rather than protein synthesis, is by the hormone cortisol. Now recall that cortisol is a stress hormone. Cortisol is released when the body is under stress, and you can imagine when we are in an extended fasting condition, approaching starvation, this is definitely a stressful situation. So under these conditions, the body is going to be maintaining us into a catabolic state by blocking protein synthesis. If we prevent protein synthesis and we allow the normal proteasomal degradation of proteins as they reach their end of their lifespan, what we are going to do eventually is shift the quantity of amino acids from the muscle protein into free amino acids. Now, by inhibiting protein synthesis, we inhibit the assimilation of these freed amino acids into protein, and therefore we now have a substantial pool of amino acids that we can be used to generate energy. Now, the first thing that we need to do before we can take this energy-rich molecule of amino acids is we have to process the amino acids and prepare it to place it into the energy-producing pathways. So let's take, for example, the amino acid glycine. Let's change that to alanine. Now, what alanine has as far as energy production is these rich hydrocarbon bonds. Each one of these carbon-carbon, hydrogen-carbon bonds contains electrons that we can oxidize and use in energy. So nature is redundant. We're not going to develop new energy-generating pathways. What we want to do is process the molecule of alanine so that we can place it into the current energy-generating pathways. So what if, for example, we take alanine and we perform a single-step reaction that takes the alanine into this structure. Notice what we've done to the alanine. We've deaminated the alanine, essentially removing the nitrogen, and we've replaced this with a ketone group. Now, if you look closely at this structure, you'll recall that this structure is pyruvate. This is also considered the alpha keto acid of alanine. Now, why is it called this? Recall that the center carbon here that's attached to the nitrogen and carboxylic acid of the amino acids is the alpha carbon. Once we remove the nitrogen and turn this into a ketone group, notice that this is a ketone on the alpha carbon, so the alpha keto acid of alanine. So any transamination reaction is going to take the amino acid and turn it into its respective alpha keto acid, because the ketone group is on the alpha carbon. Now the next question is where does this nitrogen go? We're going to control our nitrogen reactions, and so what we're going to do is we are going to perform what's called a transamination reaction. And a transamination reaction is going to be the first reaction that processing occurs for amino acids to put it into the energy generating pathway. So this means we need to take the nitrogen off of here and place it somewhere. Where we're going to put this nitrogen is on another alpha keto acid. And this alpha keto acid is going to be specifically one that we recognize. It is a 5-carbon intermediate of the TCA cycle. Recall this is alpha keto glutarate. So alpha ketoglutarate is going to be what we consider our reservoir of nitrogen. We are going to place the nitrogens of all of our amino acids that we undergo this transamination into their alpha keto acid. We're going to place all of these nitrogens on alpha ketoglutarate. Now, where specifically are we placing the nitrogen? We are placing it on, the alpha carbon. So when we do that, here is the structure that we generate. I think you'll recall this structure. This is glutamate. So by this simple transamination, by moving the nitrogen onto the alpha keto group of alpha ketoglutarate, we make this into glutamate, and in so generating pyruvate from alanine. Now, once we perform this reaction, where does the pyruvate go? Well, very simply, the pyruvate enters the mitochondria, is processed to acetyl-CoA, recall by the pyruvate dehydrogenase enzyme, and then the acetyl-CoA is placed into the TCA cycle to generate energy. So we are taking our amino acid and generating an intermediate of an current energy generating pathway, and then oxidizing these rich carbons to extract these electrons to generate energy. Now the question is here, the next question is, as all of our amino acids are undergoing these transamination reactions to prepare the alpha keto acid for energy production, we are unloading these nitrogens onto alpha ketoglutarate. But isn't this dangerous? Because this is an intermediate of the TCA cycle. So if we run out of alpha ketoglutarate, the TCA cycle will stop. And that would be counterproductive to our energy generation. So that means that we need to be able to return the glutamate to alpha-ketoglutarate. So how are we going to do that? Well, let's go backwards in this cycle. And that means we need to transfer this nitrogen onto the alpha-keto acid of another molecule to generate an amino acid. And what we're going to do here for that is that we are going to transfer that nitrogen onto oxaloacetate to generate aspartate. So oxaloacetate is the alpha-keto acid of aspartate. So let's take a look at that structure. Recall, oxaloacetate is a 4-carbon molecule. Starts out with a carboxylic acid, has a ketone group, a CH2, and another carboxylic acid. Now, in this particular reaction to regenerate the alpha-ketoglutarate from glutamate, we are going to be taking this nitrogen and placing it onto that carbon, replacing that ketone. And what we're going to generate is this molecule. So this is oxaloacetate, and this is aspartate, aspartic acid. Again, this is another transamination reaction that's performing this. Now, we might take our argument one step further and say, well, we are using oxaloacetate in order to regenerate alpha-ketoglutarate to keep this transamination cycle going of all of our amino acids into their respective alpha-keto acids. But what happens now if we generate lots of aspartic acid to perform this regeneration and deplete oxaloacetate? This will deplete an intermediate of the TCA cycle. And won't we be in the same problem as depleting alpha-ketoglutarate in the previous reaction? And the answer is yes. So that means we need another reaction here to regenerate the oxaloacetate in order to keep the cycle going. So this reverse reaction is going to be done by a special enzyme called spartyl. Once we have the aspartate enzyme, the amino acid made, the special reaction we are going to be putting this into is generating urea. So the nitrogens of urea, and we'll look at this a little more closely, will be placed onto urea by the urea cycle. And we'll look at this in a later video. So these nitrogens here of aspartic acid will be used to generate urea. Once we generate this, then this is expelled and secreted in the urine. And in doing so, we've regenerated oxaloacetate. Now, let's take a little closer look at muscle activity of nitrogen in the fasting state. Recall the job of muscle is to generate ATP. So the purpose of the next discussion is to look at the flow of nitrogen. Now, the reason we need to process nitrogen is because nitrogen is a very, very electronegative ion atom. Because of this, those electrons that are shared between nitrogen and the hydrogen are not possible for us to oxidize and extract these for energy production. And so that means any type of nitrogen-hydrogen bonds are going to be of no value to us in generating energy. And so that means that the amino acids that we are going to be using in muscle to generate energy need to all be transaminated to remove these nitrogens, to get rid of the nitrogens, so that the muscle can have that rich hydrocarbon backbone to put into the energy generating cycles. So the primary flow of nitrogen in muscle is we are going to take our amino acids and we are going to convert these into their respective alpha keto acids by this transamination reaction. And each amino acid has its own transaminase enzyme. And the first flow of information that muscle is going to do is it's going to be shifting all of these nitrogens onto pyruvate. Now recall, if we shift the nitrogen from an amino acid onto pyruvate, the pyruvate will automatically become alanine by the transamination reaction. Now what is the muscle going to do with this? Well, the alanine of muscle is going to be placed out into the blood. Once it's in the blood, the liver is going to pick up the excess alanine, and then the liver will take the alanine and convert it back into pyruvate. Now, why does the liver take up alanine avidly in the fasting state and convert it to pyruvate? Because recall, in the fasting state, the liver is performing gluconeogenesis, which simply means that all of this extra carbon source, the liver is going to be making glucose. Now, the alanine glucose conversion between muscle and liver is called the alanine glucose cycle. Now, being a cycle, that means there needs to be connection. And what does this mean is that the glucose that is being made by the liver from this alanine being secreted by the muscle is going to go back to the muscle. And the muscle will take this glucose up and oxidize it into energy. So why is this such a good deal for muscle? Now recall that muscle is losing a 3-carbon intermediate of energy production, namely pyruvate. The pyruvate is converted into alanine, and the alanine is thrown into the blood. So the muscle is actually secreting an energy-rich molecule. This is an energy deficit. Muscle does not like an energy deficit because it needs to make lots of ATP. But by giving away a 3-carbon molecule, the liver picks it up, resynthesizes glucose, which is a 6-carbon molecule. So essentially, the muscle is using pyruvate to get rid of its nitrogen. The liver is taking the nitrogen and it's making urea to get rid of the nitrogen. And in doing so, takes that carbon-rich backbone, turns it into glucose, and sends it back to muscle. So it's a good deal for muscle. Now, what happens if we go further in our fasting? So we are processing our amino acids, but now the amount of glucose is decreasing, the amount of fatty acid is decreasing because we've extended our fast, and that means we're going to have to rely on even more amino acids to generate energy for muscle. So what that primarily means is now we can take, as we're converting our amino acids to their respective alpha keto acids, let's say that we are increasing the amount of amino acids moving in this direction. In doing so, we are converting our pyruvate to alanine, but what we can also do is we can convert, in parallel reactions, we can convert alpha ketoglutarate to glutamate. So essentially what this means is that if we increase the amount of amino acids we need to get into the energy producing pathways, we need to move more nitrogen. So we're going to capitalize on moving that nitrogen onto pyruvate like we've seen up here to make alanine. The alanine again will go out into the blood to the liver, just like we've seen before. But now we're also going to take advantage of a transamination reaction to unload more nitrogen onto another substrate, alpha ketoglutarate. Now this transamination reaction also generates glutamate, but recall on the side chain of glutamate there's also another possibility to add an amine. This enzyme is called glutamine synthetase. And what this simply is going to be doing is that we are going to be adding more nitrogen onto the glutamate to make glutamine, so that now instead of just having one nitrogen in this molecule, we now have two nitrogens. The glutamine is then sent out into the blood, which then goes to the liver. The liver is going to be picking up the glutamine, bringing it into its mitochondria. And we'll talk about this transport pathway when we talk about the urea cycle. Once the glutamine is in the liver, the first thing it's going to do is it's going to turn this back into glutamate. This enzyme is called glutaminase that releases the nitrogen. It releases the nitrogen in this form, glutaminase, in the form of ammonia in the matrix of the mitochondria. The next step of this reaction is going to be converting the glutamate into alpha ketoglutarate. And this enzyme is called glutamate dehydrogenase. This, of course, being a dehydrogenase, is going to be a redox reaction. So in this process, we are going to be releasing more nitrogen as ammonia. But we are also going to be generating NADH because it's dehydrogenase. Now, once these two nitrogens are available, these two nitrogens will be used to make urea. So once again, the liver is going to be processing the nitrogen in the body to get rid of it as urea. The alpha-ketoglutarate has a specific path. This is a rich carbon source, and because, again, we are in a fasting state in the liver is going to be performing gluconeogenesis to make glucose. And once again, the glucose goes back out into the blood, and it's available for muscle.